Okay, good morning, Shafia. Good morning. Good morning, Saima. See, in the unit 4, I mean to say in the immunology, we are left with three topics. That is the autoimmune diseases. Good morning, Akshay. Good morning, Priya. We are left with the autoimmune diseases. We are left with the vaccines, and we are left with the B cell development. Okay, so these three are not greatly important topics and more not more commonly asked in the exam, also not more frequently asked in the exam. So that's why we left with. So you also no need to cover those three topics. Whenever you find the time, just have a look of those three topics. That's all. You no need to stress on that. Okay. So we have done with the cancer. We have done with the cell signaling, and we have done the all important topic of the immunology. Okay, we have 8:30 class also. Okay, and we are left with the two topics, that is the cellular communication, and another very important topic, that is the host pathogen interactions. Both topics are extremely important. For what purpose? Both topics are extremely important for the part C questions especially. Good morning, Samaya. Continue. Okay. So as I am saying to all of you, once again listen, there are three topics are left in the immunology. They are not greatly important and not oftenly asked in the exams. Okay. So now we are going to finish the two left concepts in the unit 4, that is the cellular communication, host pathogen interactions. Okay, and we have 8:30 class also. We will move to the developmental biology. Okay, let's continue the chapter of cell communication. Friends, we all people know that in this cell communications, we get the part B question also, as well as the part C question also. Okay, and the host pathogen interaction and cellular communication. Good morning, Brunalini. The advantage with these two concepts are, here less to understand, more to remember. Lot amount of the information, friends, you people has to remember every single time. More information you remember, more easily you can answer the questions. If you look into any kind of a paper from these two topics, cellular communication and host pathogen interaction, we will get, I am talking an average we will expect 8 to 10 marks. And in our 2022 paper, we got 8 plus 6, 14 marks from these two subtopics. Cellular communication we got 8 marks, and host pathogen interactions we got one part C and one part B. So total and average we got 14. I am requesting every one of you, do not skip anything in this cellular communication as well as the host pathogen interaction. Try to read everything. Okay, and don't leave it. And moreover, these two are not a difficult concept. Only the problem is that you have to do the multiple revisions and you have to remember it for the long time. That's all. But without leaving anything, please concentrate on everything. Okay, now let's understand cellular communication. So friends, what exactly we have to read in this particular cellular communication? Everyone listen to me very carefully. In this cellular communication, we have to read all the type of chemo molecule. This is one subpart. And we have to read about all the type of junctions. This is the second part. And the third and most important part, we have to read all the type of the ECM protein. These three together, we will be calling it as this cell communication. Okay, now do you think cell communication is an important process? Of course, yes. So friends, listen everyone carefully. If the process of cell signaling wants to occur, if the process of cell division wants to occur, if the process of cell differentiation wants to occur, at final, even the cell death wants to occur, Friends, it is compulsory. The cell communication is very important. So what I am saying, friends, for the cell division, for the cell differentiation, even for the cellular death, that is what we can call it as apoptosis. For every single process, what do we require, friends? We require the cell communication. Now the point is here, how one cell is going to be communicated with the another cell. See, there is no doubt about it. If the cell wants to perform their function, it has to be communicated. But you know, the cell can't communicate it directly with the another cell. The cell to cell communication can be mediated by various type of adhesive molecule. Because friends, we all people know very well, when listen to me, it's a kind of rigid topic, but we have to read with very patiently. All the cells come close together, then only they can form the tissue. And we all people know that when all the tissues come close together, they can form the organ, and when all organs come close together, they can form the organ system. Finally, an organism is getting formed. So the point is very clear. If an organism wants to be formed, from where the communication should start, that is the point. Everyone listen. If the organism wants to be formed, friends, the communication should start from the cellular level only. So one cell can be connected with the another cell with the help of a adhesive molecule. Not only that, cell not only associated with the another cell, but cell can also associated with the extracellular matrix protein. Kindly do not get confused with that particular point. Now listen to me very carefully. Whether the cell is associating with the cell, whether the cell is associating with the ECM proteins, what do we require? We require the adhesive molecule. You all people kindly look into the picture very carefully, friends. All of you look into this. This is one cell, is it? And this is the second cell. Friends, you can see the picture very carefully, and we can also know that these two cells are forming the same tissue. So it is compulsory, friends, these two cells has to be associated with each other. So how these two cells are associating with each other? With the help of camo molecule. You can see the picture over there. So that is what we can call cell adhesion molecule. And I told you, cell is not only associating with the cell. Please kindly look into the picture. See, this is the cell, and the cell is associating with the ECM also. Once again, what they require? Of course, they require the adhesion molecule compulsory. If the cell wants to associate it with the cell, and if the cell wants to associate it with the ECM, ECM is made up of different type of proteins. We're gonna read it. But currently we are talking about the CAM molecule. That is the cell adhesion molecule that plays a key role between cell to cell as well as the cell to ECM also. Kindly do not get confused with that. Now listen to me very carefully, friends. Why the communication has to occur? As I already told you, for the division, differentiation, for apoptosis, even, friends, listen to me carefully. If our embryonic cell wants to be migrated, you all people know very well, let's consider our WBC cell. Everyone, let's consider our WBC cell. Our WBC cell has to be migrated every single time, because you know very well, WBC cells are not restricted to one particular area, is it? WBC cells are keep on moving in the blood circulation. Whenever the damage occur, it entered into that particular damaged area, and it will be involved in the process of wound healing, is it? So friends, how this particular WBC cell is contacting the other cell, how it is communicating with the other cell? Of course, with the help of CAM molecule only. So friends, there are many type of or many families of CAM molecules are there, friends. But among all, what are the most important type of the CAM molecules are these four. I want you to remember every single CAM molecule without fail. Cadherin, selectin, Ig superfamily CAM molecule, then followed by friends integrins. These four are the four major type of the CAM molecule. So what kind of questions we can expect from this particular four type of CAM molecules? Of course friends, the first one is we are going to expect the question from the structure, and the second most important, we can expect the question from the function. These are all the only two ways that we can expect the question from this particular CAM molecule. So you people try to concentrate on the structure very clearly. Structure in the sense structural properties, which we can expect the question in the form of a statements. Now all the students listen. There are total three major domains are there, and friends, these three major domains are common for every single CAM molecule. These three are very common. Look into the picture very carefully. Listen to me. This is called as a CAM molecule, right? What are the various type of domains that it is having? You can only see the picture very carefully, friends. It is having one extracellular domain, and it is having one transmembrane domain, and it is called having one cytosolic domain. Once again, listen to me carefully. It is having one extracellular domain, one transmembrane domain, and another one we can call it as friends another cytosolic domain. Kindly do not get confused with this particular point. And friends, this is cell number 1. Please listen my statements very carefully without confusion. And this is the cell number 2. Of course, cell number 2 also having CAM molecule. Now that CAM molecule also having extracellular domain, transmembrane domain, then friends followed by the cytoskeletal domain. We all people commonly know that. Now the point what you people has to remember here is that, listen to me carefully, how one CAM molecule can be associated with the another CAM molecule. That is the point I want you people to remember. Everyone keep it in your mind, friends. One CAM molecule can be associated with the another CAM molecule all the time with the help of extracellular domain only. This is the most important point I want you people to remember. Listen to me carefully. Two CAM molecules associated with each other on a two different or a two same cells, mostly on the two same cells with the help of extracellular domain. And friends, listen to me carefully. When it comes to the cytosolic domain, everyone keep it in your mind. The cytosolic domain of the CAM molecule always associated with the cytoskeletal elements. So what are the cytoskeletal elements? Of course, actin filaments, tubulin filaments. So all the cytosolic domain can be associated with this particular cytoskeletal element. But how? With the help of a specialized proteins which we can call it as a linker protein. Sometimes friends, instead of using the word linker protein, we can also use the word adapter protein. Kindly do not get confused with this particular point. We can call linker or we can call adapter protein, which are connecting the cytoskeletal domain of the CAM to the different cytoskeletal elements. Very, very simple point it is. Now friends, all the four type of CAM molecules are divided into different types based on what? Of course friends, based on their interaction. What do you mean by the word based on the interaction? Whether, listen my statements carefully, whether one CAM molecule is associating with the same type of a CAM molecule, or whether one CAM molecule is associating with the different type of a CAM molecule. So based on their interactions, we categorize the four CAM molecules under two categories. One we can call, friends, homophilic interactions. Another one we can called heterophilic interaction. Name itself very clearly suggesting to all of you. Homophilic interactions are those type of interactions through which same type of chem molecules are associated with each other on the cells. Everyone listen. This is cell 1. This is cell 2. Now on the cell 1 there is a cadherin is present, one type of chem molecule. On the cell 2 also cadherin is present. Now you can see the picture very clearly. The two cadherin molecules are associated with each other with the help of extracellular domain. This is what exactly, friends, we can call homophilic interactions. Kindly do not get confused with this particular point. Next one, friends, Ig superfamily chem molecule. Kindly do not get confused that when I use the word Ig superfamily chem molecule means they are not the immunoglobulins. Do not get confused here. Their structure look like an immunoglobulin. That's why we can call it that particular chem molecule as immunoglobulin superfamily chem molecule. And anyway, this is the cell 1 and this is the cell 2. You can see the picture very clearly. Both kind of a immunoglobulin superfamily chem molecule with the help of extracellular domain they are getting associated. But the point is here, whether it is the cadherin or whether it is the immunoglobulin superfamily chem molecule, both are associated with each other with the help of homophilic interaction. I mean to say both are associating with each other. But heterophilic, see, integrin and selectin. So integrin is not at all associating with the another integrin. In the same way, selectin is also not at all associating with the another selectin at all. So all the students pay attention. Here integrin molecule associated with what, friends? Fibronectin. What is fibronectin and where the fibronectin is present? Fibronectin is present on the matrix, because it is one of the most important extracellular matrix protein. Kindly do not get confused with that. Hence we never called it as it is showing the homophilic interaction, because it is reacting with the different molecules which are of not same type. Same way you look into this selectin. Selectin, instead of associating with the another selectin molecule, selectin is getting associated with what? Friends, everyone pay attention. The selectin is getting associated with the sugars. Kindly do not get confused with this. So we can easily conclude that. Listen, friends, carefully. When chem molecule associated with the another chem molecule, then we can call it as the homophilic. If the chem molecule, instead of associating with the another chem molecule, if it associate with the any other specific molecule, then we will be calling it as, friends, heterophilic interactions. Kindly do not get confused with that. Now what we people has to study for the every molecule, as I told you, friends, we have to read the structure, we have to read the function, and I want you people to remember whether the chem molecule is calcium dependent or not. I want you people to remember even this particular point also without fail. Okay, now let's start with the first one, cadherin. You know, friends, I can say that the cadherin attachment is very stronger. It's like a velcro. This is called velcro. You know, we can see our wallet and we can see certain bags. The attachment will be like this. Correct now? Very, very stronger attachment. You know, right? When we want to open this, what we have to do? We have to apply some pressure. We have to apply some pressure, then only we can open it. Very, very easily we cannot open. Same way, friends, when the cadherin molecules are associating with each other, they will be associated with each other very, very strongly. And I want you people to remember, if the most important point, the significant point, if one cadherin molecule wants to be associated with each other very strongly, there should be a compulsory requirement of calcium. So the straightforward point you have to remember is cadherin is the calcium dependent. Kindly do not get confused with this particular point. And the cadherin molecules plays a key role in the cell-cell adhesion, cell signaling, and they play a critical role in the cell migration also. Do not get confused, friends. Functions you have to remember. All the students, please kindly look into the picture very carefully. This is cell 1, this is cell 2, correct na? This is what domain? Extracellular domain, and this is the extracellular domain, this is the transmembrane domain, this is the cytoplasmic domain. You all people very clearly observe the picture. The cytosolic domain is connected to the cytoskeletal element. You can see that very clearly. And friends, how it is associated with the cytoskeletal element, that is most important point. It is associated with the cytoskeletal element, friends, everyone pay attention, with the help of adapter protein, with the help of linker protein. Now you all people can see, friends, two extracellular domains are associated with each other. Yes or no? Of course yes. Two extracellular domains are associated with each other with the help of calcium. That's why you people has to remember a statement that cadherins are the calcium dependent molecules. Kindly do not get confused with this particular statement. And friends, there are three types. Not only three types, friends, there are many type of CAM molecules are there. I am talking about, listen to me, don't confuse. There are many type of cadherin molecules are there. But you know, based on the tissue where they are very widely expressed. What, friends? Based on the tissue where they are very widely expressed, we categorize the cadherins into different types. That is E, P, N. And friends, the point you have to remember, we categorized into these three. See, I am not saying that there is only three type of cadherins. No. There are many type of cadherins are present. But based on the expression at the higher rates, based on their high expression rates, there are three major classical cadherins are there: epithelial, placental, and neural, which are majorly expressed on these particular tissues. And friends, look into this particular specialized statement. Everyone pay attention. Cadherins join the cells of similar type by binding to the same cadherin on the nearby cell surface. What do you mean by that? Everyone listen to me very carefully. This is one cadherin, this is one cadherin. And the point is very clear. This is one cell, this is the second cell. The point you have to remember, friends, these two cells belongs to the same type. What do you mean by that? Which means these two cells are making up of the same tissue. An example, this cell is making different tissue and this cell is making different tissue. Friends, in that particular case, the cadherin molecules never associated with each other. So the statement is very clear, right? The cadherins are mediating the homophilic interactions we have studied, which means the cadherin present in one cell will be connected to the cadherin present on the same group of the cell. What do you mean by the word same group of cells, which are basically going to form the same kind of tissue only? Do not get confused with that particular point. Now you can look into the picture very carefully. If you observe the structure, extracellular, transmembrane, cytoplasmic. In the picture you can very clearly observe that transmembrane domain and the cytosolic domain. They are extremely smaller domains, and the only domain which is extremely larger is the extracellular domain. Kindly do not get confused with that particular point. Which is the larger domain, friends? Only the extracellular domain is the larger domain. Kindly do not get confused with that particular point. Friends, you all people can see this particular diagram. This is the extracellular domain, right? And you know what do we call it all these, friends? Listen to me very carefully. We call it all these as cadherin repeats. The extracellular domain consists of what, friends? Many cadherin repeats. And everyone remember, how does this particular one cadherin repeat can be associated with the another cadherin repeat? With the help of a calcium. So calcium is extremely important to associate one cadherin with the another cadherin. And as I already told you, only the C-terminal domain or the cytosolic domain can be connected to the cytoskeleton. That is about what, friends? Cadherin molecule. So we read the first type of a CAM molecule, cadherin, and second type of CAM molecule we are going to read is the selectin. Of course, cadherin is the one which is basically involved in the homophilic interactions, correct na? So friends, the moment you heard the name of a selectin, one common statement immediately has to strike in your mind, that is selectin basically involved in the heterophilic interaction. One of the most common point you have to remember. Second common point you people has to remember that we understand that the selectin is not associating with the another selectin. There is no doubt about it. And the point is here, selectin is associating with what proteins? All the students keep it in your mind. Selectins will be associated with the many type of glycoproteins which are present on the surface of other cells. So friends, these selectins can be associated with the oligosaccharide also. Even they can be associated with different kinds of polysaccharides also. Kindly do not get confused with this particular point. First important point, it plays a key role in the heterophilic interaction. They are associating with the glycoproteins, is it? Now friends, I hope you all people remember when we are reading cadherin, we have remembered one common point. What is that? Cadherin having three domains: extracellular, transmembrane, and cytosolic domain. There is no doubt about here also. Selectin also exactly having three types of domain. You all people can see here. And friends, in the cadherin molecule, which is the largest domain, there is no doubt about it. Extracellular domain is the large domain. Similarly, in the selectin also, the large domain is the extracellular domain and transmembrane domain and intracellular domain. And we discussed very clearly the cadherin molecule also having the cytosolic domain, which is associated with the actin filaments. That is what we people studied. And what I want you people to remember, I want you people to remember that this particular cytosolic proteins can be associated with the cytoskeleton same way with the help of anchor protein. There is no doubt about it. Okay, that is absolutely fine. You understand. What is the major point every student has to remember? You have to remember that listen to me friends carefully. The extracellular domain can be associated with what? The extracellular domain can be associated with the different type of sugars, correct na? Different type of glycoproteins it will be associating. Friends, please do not get confused with my words. Listen to me carefully. You know friends, this particular glycoprotein, what are, everyone listen, this glycoprotein are sugars, only we will be calling it as the ligands. Kindly do not get confused with this particular point. I want all the students has to look into the diagram very carefully. This is the selectin, friends. Long extracellular domain, very long, followed by small transmembrane domain, and very small cytosolic domain. You can observe it very clearly. And what is the one more important point you have to understand? Look into the picture very carefully. This is the extracellular domain, right? It is associating with what, friends? The glycoprotein. So the glycoproteins are basically acting as what, friends? Ligands. And I want you people to remember it. I told you now it is associated with many kind of sugars. What are these major sugars? Listen, friends. What are the major sugars that this particular glycoproteins will be associated? Listen to me very carefully, friends. One is the sialic acid, okay, then followed by the galactose, then friends followed by the fucose, then friends followed by the NAG, N-acetyl glucosamine or N-acetyl galactosamine. These four are the major sugars to which selectin will be associated. Now friends, listen, what are those? Galactose, sialic acid, fucose, N-acetyl glucosamine or N-acetyl galactosamine. And friends, another very important point I want you people to remember without fail. All the students look into the diagram carefully. This is what domain? Extracellular domain. Extracellular domain associating with what? Listen to me everyone. Extracellular domain is getting associated with the glycoproteins. Every one of you understand this particular point. Now my question here, I want you to remember how, listen friends, how extracellular domain associated with the glycoprotein with the help of a specific domains. You know friends, we can call it structural domain. Please kindly do not get confused here. We can call it as a structural domain. With the help of the structural domain only, the extracellular portion of the selectin will come and associated with the glycoprotein. Everyone listen my statements very carefully. What are those? What is that structural domain called as? You can that structural domain look like a lectin molecule in certain selectins. In certain selectins, the structural domain look like a EGF, epidermal growth factor like domain. It is looking like that. So that's why we are calling it as the EGF like domain and we are calling it as the lectin like domain. Friends, if you quickly recall, listen to me. There are many type of cadherin molecules are there. There is no doubt about it. Many types are there. But friends, among the many types, there are only some type of cadherins are widely expressed on the tissues, that is epithelial, placental, neural. Similarly, there are many type of selectins are there that are associated with the extracellular protein, associated with the glycoproteins with the help of a structural domains. But among all, the major type of selectins, friends, E-selectin, P-selectin, L-selectin. And I want you people to remember for the exam how we categorize them into different type of selectins. The answer is very simple. Listen to me very carefully. Where they have been widely expressed. Where they have been widely expressed. Based on that only we categorize it into E, P and L. Endothelial cells, platelets and endothelial cell, leukocyte. Wherever they expressed, we have given that particular name. And I want every one of you has to listen one of the most important statement, very important statement. They can ask 2-marker part B question or a direct part C question in the exam. Which of the following CAM molecule plays a crucial role in the continuous WBC migration in the process of inflammation? Once again, please listen my statements very carefully without confusion. Which of the following CAM molecules are involved in the continuous WBC migration in the process of inflammation? Undoubtedly, friends, selectin as well as the integrin. See, it depending on the question, you have to give the answer. Actually, selectins also required for the movement of the WBC. Integrin also required for the movement of the WBC. It depends what kind of question they are asking. Okay, all the students listen my statements very carefully. Look into the diagram and understand the concept very clearly, friends. Don't get confused here. This is endothelial cell, okay, and the slight yellow color you can see now, friends. What is that? That is called as basal lamina. We all know that. Listen to me, friends, very carefully. Imagine there is a damage occur. Everyone pay attention, friends. There is a damage occur. Okay, this is the area where the damage occur. Okay, friends, whenever there is a damage occur, what has to be happened? Everyone listen, what has to be happened, friends? Immediately the inflammatory signals will be released. What are inflammatory signals? You know, right? Prostaglandin. Hope everyone remember the inflammatory signals. Prostaglandins, leukotrienes, interleukins, thromboxanes. These are all what, friends? Inflammatory signals. Just listen my statements very carefully. There is a tissue damage occur. From that particular damaged area, what is happening, friends? The inflammatory signals are releasing. Point is very clear, friends. Whenever the inflammatory signals will be released, what happens? That inflammatory signals will go and activate the endothelial cell. Now I want every student has to listen to me very carefully. The moment endothelial cells get activated, please listen to me very carefully. Three type of signals, three type of CAM molecules or three type of signals can be expressed on the endothelial cell. Everyone listen. Selectin will be expressed. PAF, platelet activated factor, will be expressed. At the same time, integrin will be expressed. Don't get confused. This is selectin, this is platelet activated factor receptor, and this is the integrin receptor. Once again, listen my statements carefully without confusion. Whenever there is a damage occur, from the damaged area, several inflammatory signals will be released. That inflammatory signals immediately activate the endothelial cells. Endothelial cells get expressed and they will endothelial cells get activated and they will express three kinds of a receptor molecule. Now you all people can see the picture, right? This is the WBC cell. Friends, everyone understand, on the surface of the WBC there is a receptor for selectin also, and there is a receptor for the integrin also. And every student has to listen the diagram, look into the diagram very carefully. First, friends, listen. First, listen. First, selectin receptor associated with the selectin molecule. Where the selectin receptor is present on the WBC, associated with the selectin receptor present on the endothelial cell. But friends, what is the disadvantage here? The disadvantage is, friends, the adhesion is extremely weak adhesion. Very weak adhesion. See, you understand what I am talking. See, the WBC cell has to enter here into the damaged area. If it wants to enter, it has to undergo the process called as extravasation, which means incorporating into the damaged area. That is called as extravasation. But you know, when the selectin associated with the selectin receptor, the adhesion is a very weak adhesion. Because of the weak adhesion, you know friends, the WBC keep on rolling like this. It will be rolling like this. You only try to understand if it is rolling like this, when it can enter inside. It can't enter inside. Yes or no? Of course yes. Now all the students listen my statements very carefully, friends. Whenever it is rolling, I want you to listen. The WBC associated with the platelet activated factor. Listen, the moment WBC associated with the platelet activated factor, immediately what happened? The integrin molecule and the integrin receptor associated with each other. I want you to listen the statement without confusion. If the integrin associated with the integrin receptor, first of all, WBC has to be associated with the platelet activated factor. Then integrin and its receptor get activated and the rolling will stop. There will be no more rolling because the weaker adhesion converted into stronger adhesion. With the help of that stronger adhesion, it stopped the movement. It can incorporate into that particular damaged area. That is called as a extravasation. I'll quickly explain in 1 minute. And the first point all the students has to remember is, when you heard the name of a selectin mediated WBC adhesion, always weak adhesion. Integrin mediated WBC, always remember in your mind it is the strong adhesion. Kindly do not get confused with this particular point at all. Okay, once again all the students listen. Whenever a damage occur from the damaged site or from the damaged area, certain inflammatory signals will be released. These are all the inflammatory signals. Inflammatory signals stimulate the endothelial cell. Then endothelial cell will express three things. Endothelial cell will express, every student listen, endothelial cells first express selectin, platelet activated factor and integrin molecule. These three things will be expressed. But when the WBC cell associated with the selectin, the interaction is the weaker adhesion. That's why it's keep on rotating or rolling. During the process of rolling, this particular WBC will go and associated with the platelet activated factor. When they are associated with the platelet activated factor, immediately what is going to happen, friends? The platelet activated factor stimulate the integrin and its receptor interaction. Then friends, a weaker interaction becomes stronger interaction. Then WBC stop moving and it can easily undergo the process of extravasation, enter into the damaged area and initiate the process of inflammation. This is how selectins will play a key role in the WBC migration during the process of inflammation. Kindly do not get confused with that. So we read the first chemo molecule selectin, we read the second chemo molecule, first we read cadherin, second we read selectin, and the third we are going to read integrin. Same, when you heard the name of a integrin, what is the common statement you people has to remember? Of course, integrin molecule also associating the heterophilic interaction. There is no doubt about it. Just now we read selectins are associating with what, friends? Majorly to the glycoproteins. You know friends, integrin, I want you people to remember the major point about the integrin. Integrins are the one that plays a crucial role in cell to matrix interaction, not cell to cell interactions. Always remember this particular point, not cell to cell, cell to matrix, which means friends, integrin molecule will, listen my statements very carefully, which means integrin molecule will go and associated with a particular protein present on the matrix. Is it, friends? Integrin molecules will go and associated with a particular protein present on the matrix. what is the name of that particular matrix protein to which integrin will be associated more commonly? That is the fibronectin. It will associated with many other proteins also, friend, many other protein, but majorly, I am talking majorly, friends, integrin associated with an ECM protein called as the fibronectin. Kindly do not get confused with this particular point. And friends, if you look into the structure of a integrins, everyone please keep it in your mind. Vertebrates, friends, listen to me carefully. Vertebrates are having at least, we are talking about the least, friends. Vertebrates at least having 24 integrin heterodimers. Don't get confused. Composed of alpha subunits and 8 types of beta subunits in various combinations. What do you mean by that? Which means, everyone listen, every integrin molecule, friends, having the alpha subunit and beta subunit with a variety of combination. And you know, friends, I told you now, friends, please kindly do not get confused here. I have told you that integrin molecule is associated with the ECM protein, and I told to all of you integrin especially associated with the fibronectin. It doesn't mean that integrin is not associating with the another proteins. It will associate, but majorly with the fibronectin. And I want you people to remember this integrin molecules majorly associated with the epithelial cells and the muscle cells. Kindly do not get confused with this. Most commonly with the muscle cells. The integrins plays a very crucial role in the muscle cells. Anyway, I'll explain you. Look into this. If you look into the structure, how does the structure looks like? This is the structure of a integrin molecule. And friends, what I want you to remember for the exam is, just remember that integrins are having alpha domain and beta domain with a variety of combinations. That's all. You people no need to remember the every single compartment. It's not at all required, but I'll explain to you. That's all. All the students listen. This is extracellular domain. You all people know that. And this is transmembrane domain, and this is the cytosolic or the cytoplasmic domain. We all people commonly know about it. Now everyone just listen my statements very carefully, friends. This extracellular domain can be further divided into different kinds of subdomains. Don't get confused. The large extracellular domain can be divided into small subdomains. We can call it head piece, we can call it knee piece, and we can call, you can call knee or thigh piece, and tail piece. If you can remember, remember. Otherwise, you no need to remember at all. Don't worry. Okay. What does this particular headpiece contain? Headpiece contain propeller and hybrid. These are all the name of the proteins. Thigh protein, PS1 protein. Tail piece contain COF1, COF2 in the alpha domain. EGF repeats in the beta domain. If you can't remember this, friends, please leave it. There is no necessity to read and remember. What you have to remember is that integrin molecules contain alpha and beta subunit. One integrin, just listen, an average I am talking about. One integrin contain 18 alpha subunits, 6 to 8 beta subunits. In a different combinations it will be present. That point I want you people to remember. And the statement is very clear, right? I have told you that integrin molecules majorly associated with what? Integrin molecules are majorly associated with the ECM protein. Friends, please listen my statements very carefully. Do not get confused. Listen to me carefully. when alpha and beta, please listen, when alpha and beta associated with each other very tightly, then only this alpha beta can associated with the cytoskeleton, also can associated with the ECM protein also. Once again listen my statement. Whenever alpha beta domain, see this is integrin, okay, this is alpha, this is beta. When alpha and beta associated with each other tightly, then only these two together can associated with ECM and cytoskeleton both. You only look into the picture, every one of you. Look into the first diagram. Here the affinity is very low, which means alpha and beta are not associated with each other very tightly. Hence, they cannot associated with ECM protein. They cannot associated with the cytoskeletal protein. Every student listen, don't get confused. Listen my words. Now the affinity is the affinity between the alpha and beta is intermediate affinity. Listen to me, friends. When the affinity is the intermediate affinity, you know friends what happened? Because all the students look into this diagram, because of intermediate affinity, it is only associated with the cytoskeleton. ORR, because of the intermediate affinity, it is only associated with the ECM, but not with both. Friends, whenever alpha and beta having higher affinity with each other, now you look into this, the affinity is high, then integrin associated with ECM protein also, integrin associated with the cytoskeletal protein also. So one point, friends, it depends on the affinity between the two two integrin alpha and beta. It can associate with both ECM as well as the cytoskeleton. Do not get confused with this. Okay. So those are all the three type of CAM molecules. Remaining thing we will continue in the tomorrow class. Okay. Is it clear, everyone? Please let me know if you have any kind of a doubt. Okay. I'll share the link now. Please join at sharp 8:30 AM. We'll continue the developmental biology. Hope it's clear. You can ask me the doubts, friends. Is it clear, everyone? Quick. Please join at sharp 8:30 AM. Okay then. Perfect. I'll see you at 8:30 AM, friends. Please join 8:30 AM.